Hi everyone, good morning, good afternoon, and good evening from whichever time zone you're in. Thank you so much for joining our webinar. I'm Anushka Parashar. I'll be the moderator or the host for this event. I'm a 17-year-old high school senior from India. I'm also a theatre enthusiast and like to spread awareness through my theatre performances with my team. Unfortunately, due to the coronavirus, my team and I have not, do not, are not allowed to meet anymore. So I'm trying whichever opportunity I get and trying my best to still spread awareness and have conversations with my friends and family. I am also an executive team member with The Game Changers, and I'm also the PR and media head. The Game Changers is a one-step platform for established as well as aspiring volunteers, activists, non-profit founders, and aspiring social entrepreneurs. We want to serve as a facilitator for you to connect and collaborate with like-minded peers all over the world to create positive impact. We strongly believe in and encourage cause-based leadership and leading by example, educating the general public on important issues and encouraging positive action. Today we have with us Veni, Dole, and Puloma Bishnu, rising high school seniors from Andover, Massachusetts, USA. They have spent the last year and a half developing their research project on utilizing enzymes for plastic degradation. With the goal of reducing plastic pollution in our oceans, they have designed a plastic water bottle that incorporates engineered bacteria to degrade the bottle when it inevitably reaches our oceans. Currently, they are looking to conduct proof of concept experiments with the enzymes PETAs and leaf compost cutinase. They have presented their projects at events like the MIT Global Bio Summit, BioBuilder Final Assembly, Learn Launch Learning Innovation Showcase, Biotech and Genetic Revolution series at the Boston Museum of Science, and were the top 10 finalists at the International Spellman HVE Clean Tech competition and was selected to travel to New York City. Congratulations. They have also published their work in the BioTreks Synthetic Biology Journal and have another upcoming publication in the fall. They are passionate about climate action as well as promoting synthetic biology education and love meeting new people. We are so glad to have them with us today. And today's session will also be recorded. And at the end of it, all attendees will get a recording of our webinar and also a game created by Veni and Puloma. Veni and Puloma, take over.
Hi everyone, thanks for joining. Our names are Veni and Puloma. We're high school seniors from Andover, Massachusetts, which is about 20 minutes north of Boston in the US. So we're going to be talking to you today about our research on engineered bacteria for ocean plastic degradation. But we're also going to be giving a quick introduction to synthetic biology and letting you know how you can get involved in the field. So before we start our presentation, we're going to launch a quick poll. Oops, that's not the right thing. Okay. Sorry. So if this has popped up, if you guys could please answer the poll, and we're hoping to see a change at the beginning and the end of the presentation. I'm just gonna wait a couple seconds for that, and then I can share the results. All right, they're coming in. All right, so I'm gonna end the voting. So it looks like it's actually a more positive outcome than we'd expected. So it looks like most people are still unsure about how to feel about GMOs. So GMOs stand for genetically modified organism. And so we hope that we can get a couple more people into the support category by the end of our presentation. So to start off, what is synthetic biology? So synthetic biology is a relatively new field, but it's one that shows a lot of promise for the future, and it's constantly growing. It can be defined as creating or redesigning biological systems for useful purposes by engineering them to have new abilities. So that's a lot of words, so we're going to break it down a little bit. It starts with the central dogma of biology, which is the idea that DNA, which is the genetic code for all organisms, is converted through the process of transcription to RNA, and then through the process of translation to proteins, which carry out functions in our bodies. And so by manipulating this central dogma of biology and putting in the DNA and the RNA that we want, we can create whatever protein we want to create. And the organism will do it for us, which is great because it's a very complex process. And so rather than having to completely recreate the entire process, we can manipulate organisms into doing it for us. So there are a couple vocabulary words that'll better help you understand the presentation of our project itself. To start with, a plasmid. A plasmid is a circular piece of DNA, and bacteria naturally take up plasmids in the environment because they often contain genes that are helpful for their evolution. And so a transformation experiment is how we get bacteria to take up the plasmid that we are feeding it, or the DNA that we are feeding it. And so by heat shocking and cold shocking the bacteria, we can make its cell membrane permeable, and the bacteria will then take up the plasmid and start expressing whatever DNA is on it. Lastly, a functional assay is what you do after you've engineered an organism. It's when you test the product or, say, the protein that you've engineered the organism to make. You'll see an example of this later in our presentation.
So our project aims to focus on one of the biggest problems on Earth, plastic pollution. So the use of plastic products are not environmentally responsible. They make up an estimated 5.25 trillion pieces of plastic, and they make up to 60 to 90% of all marine debris studied. And sea turtles and seabirds often mistake floating plastic bags for food, and they can suffocate or starve. Additionally, plastic takes an average of 1,000 years to decompose, meaning every piece of plastic ever produced is still on Earth. So, considering the widespread use of plastic, it's unrealistic to expect their complete discontinuation. Therefore, we have come up with a design that maintains the convenience of a plastic bottle while also being environmentally sustainable. So we call the solution Bottles Be Gone, a self-dissolving bottle, and by placing inactive engineered bacteria under its label, the standard PET water bottle would degrade into environmentally friendly compounds upon reaching the ocean. So as you can see in the video that we just presented, we have modeled how a bottle with our technology would look like when degrading in the ocean. And just to clarify, PET is a type of plastic. It's commonly used in everyday life. So take the average Poland Springs or Dasani water bottle that's made out of PET. So it's important to note that we wouldn't be inventing a whole new bottle, but we are introducing a technology that companies can implement under water bottle labels and pre-existing bottle designs. So how would a bottle with our bacteria be designed? So take the average plastic water bottle, so like I just mentioned, like a Poland Springs water bottle. The label side would be increased to cover the whole bottle to be a full-length label. So underneath that label would be a thin, airtight film of plastic, and underneath that film would be our engineered plastic degrading bacteria.
So the bacteria we would use is called Bacillus tiansheni. This is a spore-forming marine bacteria, which makes it optimal for our design. So the ability to form spores means that in less than optimal conditions, the bacteria renders itself inactive. So it won't be taking in anything and it won't be putting out anything. So, and because it's a marine bacteria, its ideal conditions are in the ocean. And so therefore, when outside of the ocean, the bacteria would put itself in a spore state. So this means that while the bottle is in use, the bacteria will not be producing anything to degrade the bottle. But then if the bottle were to be thrown into the ocean, the bacteria would germinate, which is to become active, and it would start producing an enzyme to degrade the bottle. So in order to engineer Bacillus thienscheni, we would conduct a transformation experiment, as we mentioned earlier, using a plasmid pictured here. So this is a plasmid that we have designed with the gene to get B. thienscheni to degrade the plastic bottle. So taking a closer look at what a plasmid is and how do you design one. So there are certain puzzle pieces that you have to select and piece together. The first piece is a promoter, which is a sequence of DNA which proteins bind to initiate transcription and make the DNA into RNA. So basically it's the start sequence. Next, you have a ribosome binding site, which is the signal to the cell to start translation, which is to make the RNA into a protein. After that, you have the coding sequence, which is the actual gene that you want the bacteria to make. So in our case, it would be the plastic degrading gene. And lastly, you have a terminator, which signals the end of the plasmid and tells the cell that the protein has been made, and it stops there. So our plasmid would contain the gene for PETase or PETase. This is an enzyme originating from a bacteria called Ideonella cyconisis, which has been known to degrade PET. So by using this plasmid to transform B. thienscheni, our bacteria would produce PETase and it would degrade the water bottle. However, as we moved from the design to the experimental phase of our project, a few engineering challenges became apparent. First, being in a high school laboratory setting with our limited resources and experience makes it pretty difficult to conduct transformation experiments with B. thienscheni because it would take a designated B. thienscheni lab to be able to do this, which can take several years to establish and is beyond our own resources. Secondly, PETase is a relatively newly discovered enzyme, and therefore many scientists around the world are currently working to determine its exact properties and to engineer it to be more efficient. And therefore, we have decided to conduct some proof of concept experiments regarding plastic degradation. So to experiment with plastic degradation, we are planning to test two enzymes that show these degrading properties. One is PETase, as we had mentioned earlier, and the other is leaf compost cutinase, or LCC. And we would like to experiment with conditions of temperature, salinity, and pH to determine the plastic degrading properties of each enzyme so that we can see how well they would work in our chosen environment of the ocean. So since it's difficult to work with bacillus in our high school environment, we will instead use a cell-free system called BioBits to express the enzymes. So a cell-free system means that it has all the parts of a cell without the hassle of actually having to keep the cell alive. And it will still do the same processes of transcription and translation. However, BioBits, our cell-free system, might not give us as much product as we need. And so we will also do transformation experiments using E. coli, which is very, very commonly used in synthetic biology. And it's more likely to be successful in our environment in the high school than bacillus because it's much easier to work with. So we have selected parts for our plasmid when we were designing it that are codon optimized for E. coli, which means that they are meant to be expressed in E. coli. This includes a T7 promoter and terminator, which are just the names of the promoter and terminator, and a ribosome binding site that's also compatible. So the backbone PJL1 contains all of these parts. And this T7 system also ensures compatibility with the BioBit cell-free system. So therefore, we can use the same plasmid if we want to express cell-free or if we want to express with E. coli. Also important for a proof of concept experiment is the superfolder GFP and His tag that we are using with each enzyme. This tag will allow us to measure whether or not the enzyme is being produced and its concentration. So by tag, we mean it's a little sequence of DNA that we would stick right at the end of the coding sequence, so it would be produced at the same time as the coding sequence. Also, GFP is part of our tag, and this is a very commonly used part in synthetic biology experiments. It stands for green fluorescent protein. If you were to integrate a plasmid into a bacteria of GFP, it would make the bacteria glow green when viewed under a UV light. So this is a very useful tool to see if your protein is actually being produced, because if it's an all or nothing type of situation. So either both our protein and the GFP will be produced and we'll be able to see that, or we'll know that something is wrong and neither is being produced. And the His component of the tag also serves a similar purpose, and it helps us quantify the amount of enzyme being produced. So here you can see the plasmids that we have designed. On the right is the PJL1 backbone, as we had previously mentioned, with the gene for LCC inserted, and the superfolder GFP tag. On the right is a similar backbone with the gene for PETase, which is right here in the blue, instead of leaf compose cutinase. So the PETase gene that we are using is a double mutant that has been engineered to improve degradation abilities of PET, as compared to the original ideonyl cyclinases, which isn't the most efficient. So these plasmids will allow us to produce the enzymes PETase and LCC using either E. coli or cell-free. So after obtaining the enzyme, we are also going to perform functional assays with two substrates, polybutylene beta-ketophthalate, and also shortened to P-beta-KAT, and PNPA, which is an acceptable replacement substrate for PET in assays. And the reason that we'd like to use these three different types of plastics in our functional assay is because the crystalline structure of each plastic is probably going to affect the degradation abilities of the enzymes. And so we want to make sure that we are accurately measuring the degradation abilities and therefore using different substrates. So the functional assays will be used to test various pH conditions, salinity, and temperatures for the enzyme's efficiency. So this is an example of a functional assay, which you had mentioned previously on the vocabulary side. So it basically means that after we produce the enzymes, we're going to literally stick them on our plastic and measure how much it degrades the plastic. So it's almost the purpose of producing our enzymes.
So with everything we just mentioned, what are the benefits? So for producers worldwide, there have been efforts to reduce carbon emissions from plastic manufacturing plants, or to reduce or decrease the use of plastics in favor of reusable products like glass containers or metal water bottles. And plastic manufacturers consistently receive a steady revenue due to the convenience of their products, but they still receive a lot of negative publicity. And partaking in a positive effort like our bottle design to decrease plastic pollution while still retaining their biggest and highest selling product, we're able to compromise with major corporations as they will face less blame compared to their competitors for plastic pollution. This could also increase the demographic of a company's consumer base, and thus these consumer demands for a solution to plastic pollution can incentivize producers to take on this idea. Additionally, consumers in buying a product like ours would feel like they're making a change due to our bottle's environmental benefits. So our product also retains the convenience of a plastic bottle, and it's a practical solution. For families who are given the choice to buy a $40 reusable water bottle or a 20-pack of plastic water bottles for $6, it can be easy to understand the choice for plastic. And our idea can be of use because it means not significantly increasing the price for families, as bacteria is cheap to culture and it would result in an economy of scale. So theoretically, this would only mean a few cents more than the average cost of a water bottle, if that. Additionally, consumers can keep up with the trend. We see eco-friendly trends taking over all over social media, and it feels good to be eco-friendly. You know that there are direct benefits to your purchase, and there are health benefits as well. So consumers can stop consuming microplastics in fish, and in buying our bottle, they would be able to contribute to a healthier lifestyle, not just for them, but for future generations, and that's an important incentive. Lastly, things like cleaner beaches and oceans would be a direct result of someone's bottle purchase. Many people like going to the beach to relax, and they would rather not see plastic polluting the shore. And these environmental benefits incentivize consumers. And lastly, just like I mentioned, cleaner beaches and oceans, they're also good for the environment in general. So fishing and tourism industries can additionally flourish without plastic debris in our oceans breaking equipment or decreasing fish quality due to microplastics. And this idea could lessen the impact of climate change. When plastic is left in the sun, floating in the water for prolonged periods, carbon emissions are actually produced due to the heating of the plastic. So the environment benefits and improves because of consumer demands. So what do the people say about this bottle? So we surveyed 200 people, and after taking into consideration the estimated cost, benefits to the environment, safety, and more, over 80% responded that they would buy our bottle. Safety was an important component of our proposed solution, but our bacteria are not harmful as they are non-pathogenic, so if they were to be ingested, theoretically it would hold no effect on the human body. They do not activate in typical everyday environments, and they require a certain temperature, pH, and salt concentration that can only be found in the ocean. So that is also why we would like to work with the Environmental Protection Agency to get it approved for safety. And we also plan to perform an environmental impact analysis, as releasing a bacteria into the ocean can affect marine biomes in unforeseen ways. So the importance of our project is that there's not enough awareness on GMOs for people to make a well-informed decision. So just to clarify again, GMOs are genetically modified organisms, and we asked you before we started this presentation what your opinion of them were. So our project hopes to tackle this issue and educate the public on the importance and the potential of synthetic biology and bioengineering. So it's important that we utilize all the effective tools that we have at our disposal to solve important issues on our planet, and that includes plastic pollution. So that also means educating the public on how progressive genetically modified organisms can be for our society. So we've been featured on different news outlets such as Fox 25, WCBB Channel 5, as well as different newspapers. And through all of that, we aim to help inform the public on how important synthetic biology is through our research. But perhaps our favorite part of this project is how much it's taught us. We began it in the BioBuilder Club at our school, and BioBuilder is a worldwide organization based out of MIT that promotes synthetic biology education. There are chapters all over the world, including in Singapore, the US, Canada, Mexico, and more. And when we first joined the club at our school in the US, we had never even heard of the field of synthetic biology. But now we are constantly building our own knowledge as well as participating in global events to encourage other students to get involved. And over the last year and a half, we've been developing our project, and we have discovered the synthetic biology community. From presenting at conferences such as the Global Community Biosummit at MIT, to collaborating with different Harvard scientists working with PETase and Bacillus, we've learned that the community is always open and collaborative, and that's what makes this field so special. And as a rising field, we believe it's important to use our project to spread awareness about synthetic biology, as well as about environmental sustainability. So if we educate the public, we can help people make informed decisions. And it's important to emphasize our safety features and our extensive research into people's concerns. But the important thing is that this idea is possible. So at the MIT Makeathon in Boston, USA, we won second place for a game we created called Pocket Plasmids. So Pocket Plasmids is a fast-paced card game meant to teach people about the basics of synthetic biology. And if you happen to know the game Egyptian Rattle Screw, this card game is a lot like that one. So you can play this game with 2 to 6 players, and you start with a deck of 40 cards. All cards should be face down such that the Pocket Plasmids logo, as you can see in the top left corner, is facing up, and no one can see the colorful face of each card, including the person dealing the deck. So all 40 cards are distributed evenly among all the players. So everyone should have an equal number of cards, and once everyone has received their cards, they can look at them, but they can show no one else. And then you ask for anyone who has a card that says restriction enzyme to put down their card first. Then, based on the color, every card put down after has to match the color of the first card. In this case, the card is blue. So everyone can put down a card whenever they see their card is next in the sequence. So cards must be put down sequentially, and the sequence is restriction enzyme, promoter, RBS or ribosome binding site, coding sequence or CDS, and terminator. There is no rotation. Any player can put down a card, but only one at a time. For example, if Veni had a blue promoter and I had a blue promoter card, one of us has to be the first down to put down the card for it to be included in the sequence. So this card sequence that we just mentioned are the parts of a plasmid in synthetic biology, like we mentioned earlier in the presentation. And when all of the cards in the sequence have been put down, the pile of cards would be considered a plasmid that has been made. So if someone put down a blue restriction enzyme, the next card put down would have to be a blue promoter. And then the next card would have to be a ribosome binding site, and so on. So here we have a coding sequence card, and each plasmid made will include a CDS card, and each CDS card has a phrase below the bacteria's symbol. This is to indicate to the student what the purpose of the gene expressed by the plasmid is. So for example, we also mentioned this protein earlier in the presentation. We're actually using it in our project. A plasmid can code for a gene that makes the bacteria glow green. So that's called GFP. And we keep going with this sequence until someone has put down a terminator card. So after the terminator card of the right color has been put down, the first player that slaps the pile or the sequence of cards in the middle of the table or wherever you're playing it, wins that plasmid and receives all the cards put down. So the person with the most plasmids after all the cards have been used wins. And each plasmid can consist of a sequence of only one color. So in this case, if someone were to form a plasmid, you can see that the whole sequence is in only blue. So the purpose of this game is to not just try and have fun, but also to introduce students to synthetic biology, what parts go into a plasmid to insert a gene into a bacteria, the order in which these parts go to form the sequence of a plasmid, to convey the idea that a plasmid is used to insert a gene into a bacteria, and that a plasmid is ultimately used to express a gene. And lastly, and most importantly, this game aims to introduce and convey the power and the applications of synthetic biology. And like Anishka said, as a token of appreciation for all attendees, we will be providing a free printable version of Pocket Plasmids for you to play at home. So how can you get involved with synthetic biology? So as I mentioned before, we began our project in the BioBuilder Club of our high school. And just to reiterate, it's an organization that promotes synthetic biology education. Their website has many resources, including free textbooks, information on starting a BioBuilder club at your school, and educational lab kits. So the great thing about BioBuilder is if you're really passionate about solving problems like plastic pollution, or there are numerous problems on our Earth that need solving, synthetic biology has the potential to be able to provide solutions to these problems. So starting a BioBuilder chapter at your school could sort of help with that movement. And another great synthetic biology platform is called iGEM, or the Internationally Genetic Engineered Machine Competition. iGEM is an annual synthetic biology competition in which teams from around the world create projects. iGEM is a very competitive competition, but it highlights the synthetic biology community that we have talked about. Teams are often made up of scientists from different countries, at different ages, and with all kinds of different backgrounds. Furthermore, every part ever used in an iGEM project is available on the Registry of Biological Standard Parts. And here you can find a catalog of the parts with information, their compatibility with other parts, and past projects they've been used in. And so some of our parts are actually from that registry, so that's pretty cool. And this is where you can start piecing together your own plasmid. So thank you for listening to our presentation, and we encourage you to reach out to us. We love connecting with new people to help improve our world, and please feel free to contact us anytime. Thank you.
So for the end of the presentation, we are going to launch this same question and see where the responses go. Great. So it looks like we definitely did see a change. So a lot more people are now on the supporting GMO side, which is perfect. That's exactly what we wanted you to take away from this. Yeah, so thank you so much for listening to our presentation. We're going to hand it back to Anishka.
That was absolutely so impressive. Thank you so much for joining us today. We'll be going into the question and answer mode now. So if anyone has questions, they can definitely type it in the chat box. And I'll also be changing the setting to if you have a question, you can unmute your mic and ask us. So if you have any question, you can definitely unmute your mic and ask us. But we know that can be a little intimidating sometimes. So the chat mode is still available and you can ask your questions on the chat as well. We have our first question, which is how did you learn so much about this field?
Yes. So, like we said, we did not know anything about the field. We never even heard of it until we joined the BioBuilder Club at our high school. And so BioBuilder has tons of resources where they'll teach you the basics in synthetic biology and they'll provide you support as you build your own project. And so our teacher, the leader of the BioBuilder Club, is also very involved in the field. So she's become a mentor to us and she's taught us so much. And that's basically where we've learned. But we also, because it's such a new field, there isn't any one person that knows everything about the field. So we've learned how to reach out to new people. So for example, when we were trying to figure out if we can transform bacillus, we didn't know anything about bacillus. We'd never worked with it. And so we reached out to a scientist at Harvard University who conducts a bacillus lab. So entire lab devoted to studying and experimenting with bacillus. And so we got advice from him and then adjusted our project accordingly. So in that way, the best way to learn about the field, although maybe not the easiest, is to just start reaching out to people that are involved in it. And because it's such an open community, people are always willing to help out and give you, literally we've had people offer to ship us enzymes and ship us substrates that we might need. So it's a really great community to be involved in.
So someone asked, where do you plan to study after high school? So I actually don't know yet. That's a very interesting question. I just know that where Veni and I live, there's such a community for biology and education. So I know that I would love to stay in the area that I'm in right now, where I live at home, and hopefully stay in the city and try and get like an amazing education here.
Yeah, kind of the same goes for me. There's many universities around the world now that are working in similar fields, and I think the both of us want to stay involved in the field in a way, so that's kind of the direction we're on right now. But like Puloma said, Boston is a huge hub of biotechnology, so that's definitely an attractive option. Someone asks, how can one get involved or go about to support this project? So in terms of supporting our project, we'd love if you'd reach out to us via our email, which we can actually put in the chat. And I know that Z Game Changers will be sending it out later. But for you getting involved yourselves, the best way as a student is to check out the BioBuilder website and try to start a chapter at your school. And all you need is whatever requirements your school has for starting a club. You just need a student or two to do it with you and maybe a teacher. And you can reach out to the head of the BioBuilders organization or the BioBuilder Club organization via their website. And she can help you get started and give you all kinds of resources. You can learn about it and even like online lessons. If you'd like help with that, you can totally reach out to us and we can set you up with the head of the BioBuilder Club organization, if that's helpful. And then another way is to check out this iGEM website, the Registry of Standard Biological Parts, because you can literally go under the categories and pick out a terminator, promoter, ribosome binding site, terminator, and design your own plasmid to do whatever you want it to do.
That sounds great. We keep the question and answer mode open for another 2 to 3 minutes if anyone else has questions. You guys can always reach out to Vini and Puloma on their email, which they provided in the chat box. And if you guys want to check out Z Game Changers, please feel free to check out our website or check us out on Instagram, which is @Z Game Changers. If anyone has questions, that you guys can unmute your mics or ask any other question in the chat box as well.
So another question says, hard would you say it is to start understanding synthetic biology? So there's definitely a learning curve, but I think that the biggest learning curve is in terms of vocabulary, because people will start, I'm sure in our presentation, like people will start using bigger words and you might be confused, but a quick Google search will tell you the definition and it starts making sense from there. But in terms of starting to understand the field itself, quite literally the entire basis of the field is this kind of outline we've told you about, this plasmid constructed of promoter, coding sequence, terminator, all these parts. So it's a pretty simple field in theory, but not in practice. So it's not difficult to start understanding how the field works because there are so many resources out there. And if there is a project that you are really passionate about, then it's definitely a great outlet to do that. So for us, it was that I was the friend in the friend group that would always carry on paper straws and like hand them out to people and they'd stop using plastic ones. And so it was a pretty obvious problem that we wanted to solve. And so that kind of passion translated into our project when we started introducing ourselves to the field. I would say it took us about 3 to 4 months when we started our project to kind of get a complete grasp of how it works and to design our first plasmid. And we did this with all the resources of the BioBuilder Club, which are amazing. So we definitely encourage you to check that out.
Yeah, just to add on to that, like quickly, so Veni and I, when we started our project, like it was so hard for us to, understand. But I think the important thing is like if you're always asking questions, right? Someone will always be there to try and help you. You can reach out to so many people. So I think, the important thing is that you're always asking questions. And if you're really passionate about learning something, it won't, nothing is ever too hard to understand if you're passionate about it.
That was so rightly said, Puloma, that nothing is too hard to understand if you just put your mind to it and if you're passionate about it. And I think everyone at Z Game Changers agrees with that. Like anyone can creative change if you're passionate about it. So a lot of more webinars are coming soon. So stay tuned on our IG website, Instagram and our website. We'd like to thank Vini and Puloma once again, not only myself, but also everyone at Z Game Changers is so happy to have you, and we were so happy you came and did a webinar with us. Thank you to everyone who attended. We loved all the questions, and thank you to Vini and Puloma for answering all of them. I'll be ending with this, and seems like someone is typing, so we'll just wait for that. But thank you so much for everyone who attended, and thank you to Vini and Puloma once again.
Thank you so much. This is so fun. And thank you to everyone that attended. It really means a lot that you guys took time to come listen to us.
Yeah, definitely. Thank you so much. Yeah, thank you guys. We really appreciate you come listen to us.
Thank you so much, everyone. I completely agree. You guys are going to do such great things in your life. And with this, we've come to the end of our webinar. So thank you once again. You will get a recording of this webinar and also the game which Vini and Puloma made and also explained to us. So thank you to everyone who came and attended this. Thank you.